Hello everyone, welcome to another learning session with online biology tutorials. In today's class, we will be studying about genetic code. There are various scientists which are associated with the discovery of genetic code. Like, to name a few, they are Holle, Okua, Nirenberg, and of course an Indian scientist, Hargovind Khurana. All these scientists, they were awarded with a Nobel Prize for the discovery of the genetic code. When you should know about it? Just after the transcription, you should know about genetic code and prior to the translation. Because our messenger RNA was ready in the transcription. Now that messenger RNA has to come in the cytoplasm and it will do the translation. So there would be a sequence of bases in the messenger RNA. Right? And you know that messenger RNA, it is actually a polynucleotidic structure, but it is single-stranded. So here, to start with the explanation, you must know that the sequence of 3 nucleotides is known as codon, right? So we can define genetic code as the sequence of nucleotides or sequence of codons that will help us in determining the amino acid formed in a polypeptide. Okay, so sequence of 3 nucleotides will be known as codon, and these scientists, they discovered that each codon it specifies a certain amino acid. So that means by looking into the sequence of messenger RNA, we can deduce in the polypeptide which amino acids are going to form. Okay, the things will get clearer to you as I'll explain. So one thing you need to remember, that what is codon? It is a sequence of 3 nucleotides, right? And the combination of these nucleotides, they will help us know which amino acids are going to be formed. Now, we must know how to form a genetic code, right? So for that, you would be requiring to make a table, and you will be writing in a definite order, right? First you will make a square and you will make 4 rows in it and 4 columns. Okay, this is the way you need to proceed. If you will learn it, it would be very helpful, especially for the NEET students or the students appearing for any competition, right? And then here on the top, we would be writing bases. Here on the sides as well, we would be writing bases. Here on these sides as well, we would be writing bases. And we will be dividing them in rows as well. There is very important topic of mutation, and for that I must suggest everyone to learn how to make the genetic code table. Remember, you have to write bases in U, C, A, G sequence, right? This is your trick for the genetic code. You need to remember these 3 nucleotides, U, C, A, G. Okay, so I will be writing U, C, A, G. Fine. This will be U, C, A, G. And here also we will be writing like this, U, C, A, G, U, C, A, G, U, C, A, G, U, C, A, G. And this makes our skeleton ready for the genetic code. Okay, now this is going to be our first letter for the codon. This is going to be the middle letter, and of course the one here, it will count as the 3rd letter of our genetic codon. So let's get proceeded. Here the first letter is U, we will be writing U. The second letter is U, and the 3rd letter here we have to proceed first from here, second from here, third from here, and fourth from here. So here we will be writing number 1 base and it is U. So again our first and second letter will stay same in this block, and the third letter will change as C, A, G, right? So similarly you will make here the first letter is C, the second letter is U, and for this block the first and second letter will stay CU and the third letter will change as U, C, A, G. Let's learn again. Here the first letter is A and the second letter is U. So this will stay same in this block because we are being given as first letter A and second letter U for this block. So the third letter will change as U, C, A, G. Moving on to the next block, G is the first letter and U is again the middle letter or the second letter. So it will stay same like this. Okay, and the third letter will be U, C, A, G, right? So this way we will complete our table. So this is how I have completed the table for you. Similarly, you can also practice by making the skeleton first and then writing the nucleotides in it. Now remember, I told in the beginning that these codons, they will be coding for the amino acid. Fine. So we will be writing about the amino acids now. The first two, they code for phenylalanine. Remember in class 11th in biomolecules, we studied that amino acids, they can be abbreviated in 3 alphabets. So here PHE means phenylalanine. So the first two codons, they are coding for phenylalanine. The next two are coding for leucine. And these 4 are coding for leucine. These 3 are coding for isoleucine. This would be our methionine. I have put a star along with it. I will tell you the reason once we will done with the amino acids. Okay. So phenylalanine, leucine, isoleucine, methionine, and all these 4 will be coding for valine. You need to learn the amino acids in a sequence. You cannot make a change, right? Because this is the rule that was given by the scientists. So we need to follow in this way only. And these 4 are going to code for serine. And this one is serine, protein, threonine, and alanine. This one is tyrosine. And then these are the special ones. We will be defining about them when we will be studying the general characteristics. So you need to learn in this way only as I am writing. Okay. Arginine, serine, arginine, glycine, right? You can make your own trick and you can learn. Aspartic acid. Then you have glutamic acid. Here you have lysine, asparagine, histidine, glutamine, right? This way you need to learn the amino acids in a sequence. Just take it a homework of 4 days, and on each day you can learn the amino acids for 1 column. This is going to be very, very helpful when you are going to do the mutations. Fine. So this is how we should make the genetic code table. Now we are ready to understand the salient features of it. Okay. Now the salient features of this genetic code table are, it is triplet in nature. The salient features or the general characteristics, they are very, very important. Remember, they are in the form of triplet. Means 3 nucleotides are together. So anywhere in the genetic code table, if you see, then it could be CGU, it could be UUA. Everywhere the language is in the form of 3 nucleotides. So this is one characteristic of the genetic codon. Okay, the combination of codons are seen in the genetic code table, and right now we are studying the property of it. Okay, it is triplet in nature. It is non-overlapping. This is also very important point. So see, if the sequence of bases in messenger RNA is A, U, G, A, G, C. So you will read them out as AUG and AGC. You will never ever read them as AUG, UGA, GAG and AGC. This is a wrong way to interpret the nucleotides in messenger RNA. Okay, so you will be making group of 3 nucleotides very easy, and you will name it as one codon. Okay, so this is the another important characteristic of the genetic code table. It is comma-less, means once you are writing, you will write at a stretch, and if you will insert a comma like this in between, it is a wrong way to write, because there is no comma while writing the codons in a sequence. Okay, so this was our another point, and we have read 3 points till now. It is triplet in nature, it is non-overlapping, it is comma-less. Let's move ahead. Polarity. You know that messenger RNA was synthesized in the 5 to 3' direction. So if any sequence of nucleotides is being given to you for messenger RNA, obviously the polarity will be 5' to 3'. This is also very important. The next point is it is universal in nature. By universal, I just want to say that wherever in the world scientists are working upon the molecular biology and they are making a use of this table, the same codon will be coding for that specific amino acid. I mean, everyone is following the same table all over the world, so that constancy should be there in the data. Okay, so that is our one of the point, that it is universal in nature. Now, next thing is initiating codon. Initiating codon. If you remember, transcription was described in 3 steps: initiation, elongation and termination. Similarly, when we will be studying protein synthesis or translation, it will also be occurring in 3 steps: initiation, elongation and termination. So in the initiation process of the translation, someone has to become the initiating codon, means that will start the translation process. So this codon, which I have marked with the asterisk, this is the initiating codon. So AUG is the initiating codon, and that is why it has been marked by a star, right? So this is one of the characteristic. Now we are heading towards the another characteristic, and this is terminating codons. Now remember, I am writing codons. That means there will be more than one. So if you see here, UAA, UAG and UGA, they are all the ones that will halt the process of translation. As I said, translation would be happening in 3 steps: initiation, elongation and termination. So whenever in messenger RNA, UAA, UAG and UGA, they will come, they will be seen, then they will bring an halt to the process. Okay, so this is also important, the names of the terminating codons. Let's move on to the another thing, the number. Now if you could count, then they are 64 in number, right? 64 codons are there in the genetic code table, out of which 61 are coding for the amino acid and 3 codons are the ones which are terminating in nature, right? This is also very important. They may ask you, give the names of the terminating codons or how many terminating codons are found in the genetic code. So answer is 3. You can write their nucleotide sequence as well, UAA, UAG and UGA. And 61 codons are the ones which will be coding for the amino acid. So we have seen their number as well. Now I am heading towards very important characteristic feature. Board is generally asking a question on it every year, and that is the degeneracy of the genetic code and the specificity. Specificity of the genetic code is also known as unambiguous characteristic. Okay, so we will see what is a specificity first. Now if you see in the genetic code table, you can see that UUU is coding for one amino acid, and that is phenylalanine. No codon in the genetic code table has the same codon which is coding for phenylalanine. Means each codon is specific in coding for an amino acid. So if you will see for CUU, then you will find that it is coding for leucine. Okay, if you will see for AGU, and you will find that it is coding for serine, right? So each codon is specific in coding for one amino acid. This characteristic is known as a specificity or unambiguous. Okay, this is very important. Board people, they are asking you to differentiate between the degeneracy and the specificity of the genetic code. Remember, you need to explain as well and you need to give example as well. So if you will write the text and you will not give example, it will not give you full marks. Degeneracy can be seen here. See these 6 codons, if you can see, then they are coding for leucine only. So one amino acid is coded by many codons, and here for leucine you can see that 6 codons are seen, and they are UUA, UUG, CUU, CUC, CUA and CUG. Right? So degeneracy means an amino acid can be coded by many codons, but specificity is a one which defines that one codon will code for only one amino acid. One same codon will never be able to code for another amino acid. Now the aspirants of the competitive exams, they can also prepare this genetic code, like phenylalanine is coded by how many codons? Leucine is coded by how many codons? Isoleucine is coded by how many codons? So isoleucine, you can see here it is isoleucine, and how many codons are there? 3. Methionine is coded by just one codon. Valine is coded by 4 codons, right? And here you can see serine, and here also serine was there. So serine overall it is coded by 6 codons. So this way you can work out for each amino acid and you can learn this way as well. The students who are going to appear for the board examination, they must know how to prepare this table, which codon is coding for which amino acid, and of course the general characteristics or the salient features of the table are very, very important. You may get a 5-marker question or a 2-marker question, or just a differentiate between the degeneracy and the specificity. Now we will see a sequence of messenger RNA through which we will find out the amino acid sequence. Okay, before that please mention here tryptophan. Okay, that was missed. Now let's see an example wherein we will be taking messenger RNA. Fine. It would be better if we will take there only. because then you will get easily interpretation. Okay, so let's see if our sequence of messenger RNA is given, right? So might be they will give you the sequence of messenger RNA as A, U, G, and then they can give you C, G, U, U, G, U, C, C, U, and U, G, A. Now, if in the board they give you that the sequence of the given messenger RNA is this from 5 to 3 prime, deduce the sequence of amino acids in the polypeptide formed by this sequence. Okay, so what you need to find out? You need to find out the sequence of amino acids. Your first job is to make the codons. So you will group them to make them codons, right? So I'm grouping just 3 nucleotides together. So overall, how many codons we have received? 1, 2, 3, 4, and 5. Very easy. First step, we are done with it. Okay. Now we are heading towards the second step. Now see in the table A, U, and G. Where is our A, U, and G? Let me take some another color of the marker so that you can understand. See here, this is our AUG. Okay, and this AUG is coding for which amino acid? It is coding for methionine, right? So our first amino acid is methionine. Now let's move on to the second codon. Our second codon is CGU. So we will trace the same in the genetic code table, and here it is, right? So what are we going to write? The second amino acid, it is going to be arginine. Done. We are moving towards the third codon, and this is UGU. So quickly we will find out where is UGU, and here we got it. And this makes the third amino acid as the cysteine. Now let's see for this one, CCU. We will find it in the table, and here we go. We have found it. It is for proline. Okay. Here it is proline written for all the 4 codons. So that's why I have written proline. Now the last one is UGA, and here you can see that UGA is the one that will halt the process. So that means there would be no dash in the end. So we will do what? There will be no amino acid in the end. Okay, so what are we going to write? We can write the amino acid sequence now as N-terminal, because whenever we are writing polypeptide, they are always written from N to C-terminal. That's what we have learned. N-terminal is the free amino group on the left, and C-terminal means the free carboxylic group on the extreme right side. So our sequence will be N-terminal, methionine, arginine, cysteine, and then proline, and this is going to be the C-terminal. So this is the answer, and this is our problem. Okay, so we have deduced with the given messenger RNA sequence, we can find out the sequence of the amino acids. I hope this was helping to you, and you are sharing my videos with your friends. If you haven't, please do share maximally with your friends so that everyone can take the benefits of these tutorials. Stay tuned for more videos and do subscribe to my channel. Thank you.